We may not realize that we're performing a scientific experiment of sorts when we squeeze produce at the supermarket, but in a way, we are. Studying how living materials such as food or tissues stretch or squeeze when forces are applied not only help us determine if a piece of fruit is ripe, but also help us study how to limit the growth of cancerous tumors, understand muscle atrophy, traumatic brain injury, and other diseases. Measuring or quantifying how stiff inanimate materials are, such as steel or rubber, typically requires cutting a sample into a specific shape and putting it in a machine. This is impractical for measuring the stiffness of tissue inside the body. For these measurements, we need to test them locally in their native environment. We also must determine how the tissue adapts over time in response to the loads, much like how a carpet slowly recovers a dent left by a chair leg. This property is called viscoelasticity and can be described using a material model represented by springs and dashpots. Building on existing research, researchers at MIT have developed an experimental technique called volume-controlled cavity expansion, or VCCE, that is robust, minimally invasive, and allows living materials to be probed locally in exact spots without removing them from their environment. In VCCE, a syringe needle is inserted into the material. The needle is filled with water and the syringe is pushed, causing a water-filled cavity to expand at the needle's tip. A connected sensor actively monitors the pressure applied by the fluid on the material. The syringe plunger is then pushed and pulled, thereby inflating and deflating the water-filled cavity in multiple cycles that are volume-controlled. The pressure describes the forces being applied on the material, while the volume describes how the material is squeezing in response. By inflating and deflating the cavity at different controlled speeds and allowing the material to relax and recover at controlled volumes, the stiffness and viscoelastic properties are studied. The material parameters are then obtained by fitting the pressure-volume data to the springs and dashpots model. The researchers suggest that this technique can be used in the future to track variations in diseased tissues, to study how living materials grow, and for various additional applications.